right, good morning. Why don't we go ahead and get started? I'm Jessie Kennedy. I'm with marketing, and I have the pleasure of introducing our speaker today, Dr. Miriam El-Kawand. Today, Miriam is going to be featuring one of our Ask the Experts installments. We do these occasionally to bring our technical expertise to you, the community, to help you understand proximity ligation assays, Hi-C, and how we can assist you in bringing those into your own labs and progressing your research. So today, Miriam is going to speak about how to choose the right assay for your project and really demystify Hi-C and all the different methods that there are. Let's see. There's Miriam. She is our head of customer success here at Dovetail Genomics, and she's been leading our customer success team in proximity ligation kits for many years, since 2018. And she completed her PhD in microbiology in the UK at the University of Warwick. Prior to joining Dovetail, she was a lecturer in molecular biology and genetics at St. Joseph University in Lebanon. So we're very pleased to have her expertise today and answer all of your questions. Miriam really knows the ins and outs of these assays. And without further ado, I'll hand it over to you, Miriam.
Hi, everyone, and thank you for joining us. In today's presentation, I'll walk you through how to choose the right Hi-C assay for your project. So I'll start with some introduction on what is 3D genomics, what role it plays, how is it studied. I'll share with you the different Hi-C flavors, and then we will end up with a decision tree that will help you identify the right Hi-C assay based on your research scope. So let's start with what is 3D genomics. 3D genomics is that deliberate packaging of the chromatin in the nucleus. And it's based on the fact that the genome is not randomly packaged and positioned in the nucleus. And the 3D organization of the genome shows a hierarchy. So at the high level, you see these chromosome territories, which are the non-overlapping regions that each individual chromosome occupies within the nucleus. And if you zoom in more on the chromosome, you see that it's organized in A and B compartments. So these compartments, A, are active chromatin, open chromatin, that show a high level of transcriptional activity, and B is more the repressed, gene-depleted, and closed chromatin. If you zoom in more, you see this other topological feature, the TADs, or topologically associating domains. And these domains promote interactions of sequences that are contained within the same domain, and they limit interactions of sequences outside of that domain. So in a way that if you have an enhancer, it actually interacts and acts on a gene promoter that is located within the same TAD, and it limits the interaction or the ability of this enhancer to act on a promoter that is located outside of that TAD. So it helps with regulating gene expression. If you zoom in a little bit more, you see that you have these loops, and loops is actually this chromatin folding that brings together a regulatory element such as an enhancer with the promoter, and it recruits the transcription machinery, and then it activates gene expression. So why 3D genomics is important? 3D genomics plays an active role in cell biology. It can control gene expression by influencing the accessibility of the transcription machinery to the DNA. It also helps maintain the chromosome structure when you have cell division, so it prevents any breakage or any loss of genetic material. It also helps the cell respond to any DNA damage. So chromatin folding makes it easier for the repair proteins to access the damaged site or the damaged DNA and to repair it. Also, the chromatin remodeling, which is this dynamic change of the chromatin folding, is one of the epigenetic regulation mechanisms. It actually plays a role in gene expression by making it either easier or difficult for the transcription machinery to access a DNA site. So in addition to the role that 3D genomics plays in more of an epigenetics and gene regulation applications, it also plays a role in genome assembly applications. So researchers can use this 3D genomics information to infer what sequences come from the same chromosome how they're ordered, how they're oriented. And as such, this information is used to assemble sequences in chromosomes, and you can end up with a chromosome level assembly that is a platform, for several downstream studies that have implications in developmental biology, evolution, conservation, and many others. So how do you study the 3D genomics? 3D genomics is studied by several techniques, such as fish microscopy or chromosome conformation capture, 3C techniques. And for the purpose of today's presentation, I'm going to be focusing specifically on the chromosome conformation capture techniques. So this category includes different techniques that all have in common that they use proximity ligation to infer the 3D proximity of regions of the genome that are distant to each other on that linear sequence space. So they all use this core workflow of proximity ligation. And the idea behind these techniques is to capture interactions by cross-linking the chromatin in the nucleus. You'll then digest the chromatin and ligate fragments that are co-localized in the 3D space. So what you end up with is these linked DNA fragments. So you have, this is a fragment that comes from a genomic region. This is a fragment that comes from another genomic region. Those two are in close proximity in the 3D space, but they're not next to each other on that physical linear space. So once you generate this proximity ligated chimeric DNA molecules, you can reverse the crosslinks, degrade the protein, and you end up with these molecules that you can take into downstream steps. So all of these techniques have this core workflow in common, but they do have the differences in how they're detecting their interactions and what interactions they are detecting. So at its inception, the 3C technique was used to interrogate the interactions between two defined DNA sequences. So you're really establishing a 1 versus 1 relationship, and it used PCR amplification to detect that interaction. Then 4C came in, and 4C uses microarray or sequencing, and it asked the question, what are all the interactions that are actually occurring with this defined DNA sequence? So you're going from 1 versus 1 to 1 versus all, and you're getting all the interactions in the genome that are associated with your specific selected DNA sequence. 5C also uses microarray or sequencing, but now your approach is many versus all. So you're capturing all the interactions that are happening, but with more than one defined sequence, you actually have selected many defined sequences, and you're asking what are all the interactions that are associated with these sequences. Now, HiC was the first method that is genome-wide, not targeted, hypothesis-free. You're not having to pre-pick and select specific DNA sequences. You're actually asking that question, and you're able to cataloging all the interactions that are happening in the genome across all the different regions. So that is all versus all relationship. And it uses next generation sequencing after the proximity ligation in order to capture those interactions. HiChip and capture HiC, these are also methods that are using next generation sequencing, and they use the HiC workflow, but they also, they're not genome-wide, they're targeted approaches because they're actually anchoring the interactions to either a protein of interest, and that would be in the case of HiChip, or specific sequences of interest, and that would be the capture HiC. And in the next slides, I'll talk a little bit more in detail about the HiChip and capture HiC assays. For now, I'll dive a little bit more into the HiC evolution. So as I mentioned, the HiC method that was developed by Lieberman, Aiden, et al. In 2009 revolutionized how you're capturing the interactions that are happening in the 3D genome because it's hypothesis-free and you're really able to cover the entire genome. But it actually also had some limitation, and that's mainly because the chromatin was digested with this method with restriction enzyme. So you're really limited with having restriction enzyme sites. And what you end up with are sequences that are overrepresented and sequences that are underrepresented or not present in your data, because these would be regions of the genome that have few or don't have those restriction enzyme sites. So in 2016, a new method of restriction enzyme-free Hi-C was developed. And instead of using restriction enzyme to digest the chromatin, DNase was used to digest the chromatin. And by using DNase, you actually are getting a more uniform coverage of the genome. You're not limited to restriction enzyme sites, and you're not blind to sites that don't have these restriction enzyme motifs. So that was the first development. And then in 2019, a new Hi-C method was developed. And this time it actually used MNAs for digesting the chromatin. So it's still based on the same concept of proximity ligation, but the digestion of the chromatin that happens after cross-linking is now done with MNAs. And MNAs has this exo and endonuclease activities. So it can cleave the linker DNA and it can chop back until the mononucleosome or the nucleosome. So what you end up with are fragments of a nucleosome size. So you're really able to resolve all of your genome up to that nucleosome level. So around 150 base pair is how you can get, to ask your questions and interrogate interactions. So microC that uses MNAs opened up the highest achievable resolution of all the Hi-C flavors out there. What all of these Hi-C workflows have in common is that they usually take around 2 to 3 days or longer to actually complete. So you start with your sample, and after 2 to 3 days, you have your library that is ready for sequencing. So in 2022, Dovetail developed and launched the toppling assay. And this assay actually uses TN5 for the digestion or the fragmentation of the chromatin. And what it offers is this easier or quicker workflow. So you're able to start, in the morning, your library preparation, and by the end of the day, you're going to have library ready to put it on a sequencer. So you start from a sample to sequencing ready library in a day. So that's more about like the genome-wide Hi-C approaches and how they evolved. But I did mention I'm going to spend a little bit more time as well on these key considerations you're going to think through when you're making the decision, which is the Hi-C assay that is right for me. So other than, looking at how the chromatin is digested, you really need to look at what resolutions you're interested in, what topological feature you're interested in, and these things will be factored in when you choose the right Hi-C assay for you. So let's spend a little bit more time talking about that. For the AB compartments, they're usually called at 1 megabase pair resolution. So you really need a low resolution to call these AB compartments. As I mentioned earlier, the A compartments are the open and are the transcriptionally active compartments, and they're usually located closer to the center of the nucleus. The B compartment are the repressed, gene-depleted, and closed chromatin that are usually located closer to that nuclear envelope. And domains of that same compartment preferentially interact with each other. So you don't actually have A and B compartments that are contiguous on that linear sequence. You have them in that 3D space talking to each other if they're belonging to that same compartment. So in a way, when you visualize those AB compartments on a Hi-C contact map, what you're going to see is this checkerboard pattern. And what this is showing you is actually a reflection of alternating regions of high and low interaction. So let's take a look at this. What this is showing, or what this heat map is showing, is that this domain and this domain don't interact much with each other. But this domain and this domain interact more with each other, meaning that these two belong to different compartments and these two belong to the same compartment. So you're going to be able, with the Hi-C data, to start and look more at, how is your genome or how is your chromosome organized in these A and B compartments. If you wanted to look with a closer look and you're aiming for TADs, you're actually looking at 10 to 25 kb resolution. so a little bit higher resolution from what you start with for AB compartments. And the way you will see these TADs in a Hi-C map are like triangles. So what this is showing is that you have more interactions within the same TAD than you have interactions with sequences outside of that TAD. So in a way, you have these TADs that are actually constituting these boundaries between the different clusters or domains. So if you have an enhancer in TAD 1 and you have a promoter in TAD 2, this boundary is going to limit the interaction and the ability of this enhancer to act on that promoter. And usually the enhancers contained within a TAD, they interact and act on promoters that are located within the same TAD. So they're associated with that same gene regulation mechanism. For a higher resolution, you're now able to start to study and call loops. And again, the loops are this folding of the chromatin that is bringing a regulatory element in proximity to a promoter. For loops, a standard resolution would be 5 to 10 kb, but some researchers actually call loops and resolve the genome at higher resolutions. So you can see 1 kb, even 500 base pair resolution. And the way you would see the loop on a contact or a Hi-C contact map is a strong signal at the apex of a triangle. And what this is showing you is that you have this region in the 3D space in proximity or interacting with this region. And that's usually a regulatory element and a promoter. The way you would see this, maybe if you're more familiar with that other depiction of loops, is you would see them as arc on like IGV or WashU browser. And it's also linking those two regions together that are not necessarily in close proximity in that linear space. And in order to facilitate the formation of that loop, you have a lot of structural proteins that are actually, associated with this loop formation, such as CTCF or cohesin. So a lot of researchers also study loops that are associated with CTCF sites to better understand all that loop folding. But if you want to take this one step further and resolve the genome even at a higher resolution, so you're really now starting to look at things that are 500 base pair and less, this is when the question is more about where are the nucleosomes positioned in the genome and how they're talking to each other. And this is where usually micro-C and MNAs, because of this very high resolution that it offers, this is when it usually comes in to help researchers address this question. So here on this track, you can see that each peak represents the position of a nucleosome. And then here with arcs, you can see the different interactions that are happening between nucleosomes. So this is another way of using the Hi-C data beyond, the loop as a topological feature, and you're starting to ask questions around that building block of chromatin folding. So that was more with Hi-C genome-wide approaches, but you do have targeted approaches also that leverage the Hi-C workflow, but you're not interested in all the interactions that are happening in the genome. You're interested in either a protein-centric view into those interactions or a sequence-centric view into those interactions. And this is where those two options come in, high-chip assay or hybrid capture. So with the high-chip assay, what you're doing is you're starting with cross-linking your chromatin, digesting your chromatin, but before you go into the proximity ligation, you're actually doing a chromatin immunoprecipitation with your antibody of interest. And then you ligate, so you have the proximity ligation of your fragments, you convert that into a library and you sequence. And what you will end up with is, one, you're going to get like a traditional ChIP-seq experiment, you're going to get the mapping of the binding sites of your protein of interest. But unlike ChIP-seq, you're going to get one additional layer of information in that 3D space. And that would be, you're going to get all the interactions that are facilitated by your protein of interest. So all the interactions that are actually associated with your protein DNA binding sites. So you're anchoring all of the interactions that are happening in the genome to those that are specifically mediated by your protein binding, your protein of interest. And this is something you can do with this high-CHIP assay. If you're actually more interested in, things like understanding what is linked to your promoter regions, what you're going to do is more this hybrid capture approach. So the hybrid capture is where you actually complete first this high-C workflow, and usually we use micro-C is used for that first part of doing the high-C proximity ligation library. But before you sequence, like you would do if you're just interested in a genome-wide approach, you will take that library and hybridize it with probes. And these probes will be targeting specific regions of interest. Now, these regions can be promoters and they can be other, custom regions that you're interested in. So when you're hybridizing with these probes and you're pulling down, you're actually only getting the interactions that are linked and associated with your sequences of interest. And then you have your capture library that goes for sequencing. So you get the data that's interrogating interactions with specific sequences that you're interested in. So these are the more, common targeted approaches for high-C that you can use for your research. Now, a little bit more into the high-C solutions that, that are offered by Dovetail. So you can, think through these high-C assays when we get to our decision tree and decide which one is the one that is right for you. And all of these solutions are actually offered as kits or services. So you have that option available to you, but it starts with knowing which is the best suited assay for your research. So to start with OmniC, the OmniC assay uses the DNAs to digest the chromatin. So it is this a primary advantage of having that uniform coverage of the genome. And it is a genome-wide approach. So it's, like the restriction enzyme-based high-C, it targets and maps out all the interactions that are happening in your genome, but it uses DNAs instead of a restriction enzyme to give you this uniform coverage of the genome. So you're not biased to only where you have those restriction enzyme sites in your genome. micro-C uses MNAs to digest the chromatin. It's also a genome-wide approach, just similar to OmniC, but what it offers is this resolution, this highest achievable resolution, because you can start to resolve your genome up to that nucleosome level. TopoLink uses TN5 for fragmenting that chromatin. It is also a genome-wide approach, so all of those three are genome-wide approaches, but the primary benefit is that it can actually still get you to call your topological features and map all the interactions in your genome, but it does that with a quicker workflow. So you're able to complete your sample to library prep in a single day. High-CHIP and promoter capture, these are the targeted workflows. They both use MNAs to digest the chromatin. And as I mentioned, they're targeted. The difference between high-CHIP and promoter capture is what is your viewpoint into those interactions? Do you want to anchor your viewpoints into a protein that you're interested with, or do you want to anchor those interactions with a DNA sequence that you're interested in? I added this note next to promoter capture just to highlight that if you're asking for a promoter capture as the right assay for you, you're actually going to be getting micro-C as well. Because with a promoter capture, you first run a micro-C workflow and a micro-C library preparation. And that library would be your input for the promoter capture. So you use that library to hybridize with your probes and then continue on with this enrichment workflow. And as the name indicates, it's a promoter capture. So the solution we offer is primarily targeting promoter regions. And these are regions that would be, all the promoters in the genome. So that's why it's a promoter capture, not only the hybrid capture where you can customize the region of interest that you're interested in. That's not necessarily a promoter. So knowing that these are, the assays that are available to you, how do you now make the decision with what is the assay that you're going to choose for your research? And here is where we get to this decision tree. So the first question to ask is, what is the application that you're interested in? Are you interested in a genome assembly application, or are you interested in an epigenetics gene regulation application? If you're interested in genome assembly, then you really want to leverage that uniform coverage of the genome. So you can get all your sequences of the genome presented in your data set. And this is where OmniSeq comes in. It's genome-wide, and it's digesting the chromatin with DNAs. So OmniSeq would be the right kit for your genome assembly application. Now, if you're interested in epigenetics or gene regulation, you have to ask an additional question, which is, are you looking to map all the interactions happening in your sample, or are you looking to map interactions that are more associated with regions or proteins that are of interest to you? If you're looking to do all the genome-wide or map all the genome-wide interactions, either in one experimental condition, so you're just looking to discover what are the interactions that are happening in a specific experimental condition or a sample, or also if you're looking to map those genome interactions, but comparing them between different experimental conditions, for example, cancer and normal cells, but you're looking to see, at the scale of the entire genome, what are the interactions that were gained, lost, what interactions remain the same. So all of these questions are still within this genome-wide approach. You're looking at now two kits or two assays, either the microC assay or the TOPOLINK assay. To better decide on which one to go for, you need to ask the question of, what is the resolution that you're interested in, and what is the topological feature that you're interested in targeting? So if you're looking at a resolution up to 5 kb, so in that sense, you're really looking for AB compartments, stats, and loop calling, but at a 5 kb resolution, then TOPOLINK is the right assay for you, because TOPOLINK will allow you to call all of these topological features in a quicker workflow. So you actually get done with your library and have your data set to analyze quicker. If you're interested in a resolution that's more than 5 kb, so if you're interested in calling loops at a more than 5 kb resolution, for example, at a 1 kb resolution, or if you're interested in nucleosome positioning, nucleosome to nucleosome interaction, then really microC is the right assay for you, although it would take 2 to 3, usually 2 days to complete the workflow. So it's a little bit, takes 1 day longer than TOPOLINK, but it's going to offer you, because it digests the chromatin with MNAs, this highest resolution that you can achieve with lower sequencing burden compared to other high-C methods. So microC in that sense would be the right assay for you. Now, if you're looking for a targeted workflow and you have specific regions of interest, either from a promoter's region of interest or other regions in the genome, you're really looking for the question between a high-chip assay or a promoter capture or pan-promoter assay. So, to get to the right assay here when it comes to targeted workflow, you have to ask, what is your viewpoint? So, for example, if you have a transcription factor that you're interested in, it's well-defined, and you're asking the question, how does this transcription factor influence gene regulation? So you're interested in understanding, you know where it binds. You're interested in understanding what interaction are happening with this protein binding site. Then you're really looking to do a high-chip assay. So you're just getting all these interactions that are targeted with your protein of interest. In that sense, high-chip would be the right assay for you. But if you have, for example, GWAS data, and you've identified SNPs or variants, and you're trying to link them to promoters, this is where the pan-promoter assay is the right assay for you. So now you're asking the question about what are the sites or the regions that are associated or interacting with these promoters in the genome. And as I mentioned in the previous slide, when you're actually getting the pan-promoter assay, you're also doing the microC library preparation. That would be the first step in your workflow. So the microC and pan-promoter would allow you to get interactions that are specific with your regions of interest. In this case, you're asking, what is linked to those promoter sites in my sample? So, hopefully this will help you identify the right assay for you based on the question that you're asking. But also I wanted to highlight a little bit the sample types that we have for these assays. This is a question that we often get. So I wanted to use this opportunity to also talk a little bit about that. So for OmniSea, we've used OmniSea and have experience with the barnyard. So if you have a sample, that belongs to any taxa and you're looking to assemble the genome, just know that OmniSea has been kind of validated and worked on the barnyard samples. For TopLink and MicroSea has been validated on all mammalian samples, so that is blood, tissue, cells, Drosophila, rice, and yeast. So if you're interested in one of these sample types, you have the workflow ready and available. If you're interested in actually my MicroSea assay, it's still the right assay for you, but your sample type is none of those 4 buckets, then please reach out to us and we can help with how you'd go about setting up the library preparation. For TopLink, it has been mostly validated for mammalian cells. So if you're interested, for example, in Drosophila, rice, and yeast, you may want to explore MicroSea. It will still give you the topological feature you're interested in, so you don't need to sequence as much for a less than 5 kb resolution, but you have workflows that are available to you to use for the library preparation. The MicroSea and Pan Promoter, here we have the Pan Promoter panels that are either designed for human genomes or mouse genomes. So currently it doesn't support outside of these sample types. For HiChip, it's been validated for mammalian cells, but if you're interested in anything outside of the mammalian samples that you see the user guide on the website, please contact us and we can share with you how do you go about these library preparations. That is it from my end. I'm happy to take any questions or clarify any points in the presentation to help more choose the right assay for your research.
Thank you so much, Miriam. Great presentation. And just want to remind everyone, if you have a question, you can submit it to the Q&A box. Thank you.
So, starting to get some questions. Let me just read the question to you. So the first question is about the number of cells that you need for these assays. The number of cells, it depends on what is the assay that you're targeting. But for example, if you're looking for the genome-wide assays like MicroSea or TopLink, you're looking at 1 million cells as a starting point. That said, if 1 million cells are not available to you and you're working with limited samples, we do have for MicroSea workflows that support lower input. So you can actually get, down to even 10,000 cells for you as a starting point. For things like promoter capture, one feature that resonates with our assay users is the fact that for, other in situ methods, it calls for a large number of cells, but the promoter capture actually just calls for 1 million cells. So you're going to be starting first with a MicroSea library preparation, and you're going to use 1 million cells for that before you continue with the target enrichment workflow. Do you have antibodies for HiChip, or do we have to get our own? So this is a great question. For HiChip assay, the antibody itself is something that is, user supplied. If you're doing that in a kit manner, for services, we're obviously going to use the antibody that we have in-house. But I think, that the other side of the question is, what antibodies have we worked on, have we validated for our assay? So we do have a list on the website for all the antibodies that we validated. And these are some transcription factors, most of the histone marks. So this is something that we've already, validated that you can use this antibody with our assay. If you're interested in an antibody or a protein of interest that's not covered in this validated list, please reach out to us. This is something our service team is equipped to do. So they're actually they can take an antibody that's not validated and validate that and then run the high-chip assay on. Or for kits, for example, we can share with you how you can start with validation and then move on to the library preparation. That the important point usually is that you are starting with a ChIP-seq validated antibody. So this antibody would have been used for ChIP-seq experiment successfully, and then you're using it for high-chip for additional 3D data. Can I use multiple antibodies in high-chip? That's, again, a great question. So for the antibody use, for every high-chip library preparation, you're actually using only one antibody for that chromatin immunoprecipitation. And this is where, the reason it's a great question, it's because we often get that from other prospects or customers as well. So if you have, for example, several antibodies that you're interested in, then you really need to ask the question whether it would make more sense to go with several antibody high-chip preparations. So let's say you have 5 antibodies that you're interested in. Now you're asking the question, you're actually going to do 5 high-chip libraries and using for each library a separate antibody. You can't multiplex them in the same library prep. So the question becomes, does that work more and better for your budget, for the number of cells that you have available to you? Or do you want to rethink how you're approaching this question and actually try to target it from more of a promoter or a more of a genome sequence-centric view? So in a way, is high-chip the right assay for you? Or do you actually find just you're getting to that answer with less sequencing and also less cells that are available to you if you're going the route of promoter capture? So I think, it's not a straightforward answer here, but what we've seen is usually if you have 2 to 3 ChIP-seq data, then high-chip is still the right assay for you. So you're doing 2 to 3 high-chip libraries. But when you start having more than that, you really need to see what is it that you're trying to address. So I would definitely look at the supporting data you have and ask the question, is the high-chip the right thing for you, or is promoter capture the right thing for you? And if you needed more kind of offline advice, please reach out to us. I'm happy to consult more on your project. So So for microSEQ, your new protocol set 20 to 70% mononucleosome. Do you prefer lower end or higher end of 70%? Which one is better? So that's a question about the new microSEQ protocol that we released. We call it just making the best assay even better. And that is start talking about the highest achievable resolution that you can get with a microSEQ assay. And the question is more about the digestion. So the digestion with MNAs with our first earlier version used to be between 40 to 70%. Now we expanded that window to be 20 to 70%. So if your digestion is between 20 to 70% mononucleosome is what you're seeing in your you're actually able to proceed with the protocol. Now, in terms of where you want to be, we do have a white paper that I can share with you, and it explains exactly what is the impact or influence of the digestion profile on your ability to call topological features. But in brief, if you're looking to call those ultra high resolution features, so if you're looking for loops that are more than 5 kb, if you're looking for nucleosome positioning, nucleosome to nucleosome interactions, you do want to digest your nucleosome more. So you do want to be at that 50 and 60% mononucleosome. For everything else, if you're above 20% mononucleosome, you're actually good to go. Can you elaborate on the bioinformatic analysis approach for the data? Yeah, so what I can say, the time we have, that we do actually use more the open source tools that are available for, all of these, to process all of these high-C data. So it's nothing like it's nothing in a gray box or something that we have developed. It's more of the open source tools that are readily available for high-C data. And you have few options for the analysis. You actually can use some of the tools that we're highlighting in our Read the Docs pages. So for every product, we have a Read the Docs page that's available. And this tells you the tools that we use, for example, to call topological features, to call AB compartment or stats. We usually use the juicer suite. You can use eigenvector or the arrowhead. So usually the standard tools that are used in juicer. Now, this kind of field is always evolving. So you're going to see that there are a lot of tools out there, and there isn't a consensus on what tool works best for your data. But we do have tools that, we know have worked well for us. We think the results, they're easy to use and they get you to that analysis, quickly. And these are the tools that are available on our Read the Docs page. So we can share that with you. Also, we do offer the analysis as a service. So if you're interested in analyzing your data, but you want us to do that, we can do that as a service. And you also get, bioinformatics consultation services if you want something beyond the topological feature calling and more customized for your specific research. So maybe to better address this question, we do have a set of tools that we use for these high-C data processing that are available on Read the Docs. And if you're interested, please let me know and I can share those with you. Have you ever tried to apply antibody for toppling to make high-chip-like assay? That's again a great question. For now, this is not something that we're offering. So as of now, that the guidance for high chip is that it's actually using as a backbone the micro-C assay. So you're still getting the advantage of MNAs in a way that you're able to get all of these regions that are not biased with a restriction enzyme site, and you're coupling that with chromatin immunoprecipitation. For now, this is not something offered with toppling. So if you're interested in high chip, you're going to use the micro-C, workflow. But just maybe a clarification for high chip. High chip is different than promoter capture in a way that promoter capture, when you're running the workflow, you first finish the micro-C library preparation, and then you can do your hybridization. With the high chip assay, you don't need to, you're not getting, two assays, or two kits. So the micro-C steps are integrated within your high chip workflow. The way you're digesting the chromatin, the way you're running the proximity ligation. So really think about it as it's the micro-C assay, but you have the chromatin immunoprecipitation as one stage that is added to that workflow. Another question is about the depth that you need for a 5 kb coverage. So the standard, 5 kb resolution usually can be achieved with around 80x coverage. So, to put that in terms of depth, if we're talking about a human genome, you're achieving 80x coverage with around 800 million read pairs. So all of these high-C data flavors are actually going to be sequenced in paired end mode. And what you would want to aim for a 5 kb is around 800 million read pairs. That is using, either the toppling assay or the micro-C. May I know if there's a QC to check if the sample preparation worked right without having to wait till sequencing? So the way the workflow is set up is you have in the workflow some QC checkpoints just to tell you, that everything is going well. And if not, you can take a corrective measure. The final QC checkpoints when you're done with the library preparation is the final library yield. So the yield is usually an indicator of the how well, the prep worked with your sample. And then before you go into deep sequencing, you do have the option to run a shallow sequencing. The shallow sequencing is usually, like a 1 to 2 million read pairs if you're looking for those genome-wide approaches, and it's around 10 million read pairs if you're working with high chip. And what this enables you is that you're actually going to be able to run these shallow sequence data with our QC analysis workflow. Again, that's something that's easy to use, available on our Read the Docs page. So you will run that workflow with the shallow sequence data. And from the QC output, you're going to be able to assess the complexity of your library, the long-range information captured in your library. So you're going to assess the quality of that sample prep before you proceed with deep sequencing. Some users like this option, before kind of investing in deep sequencing. For the shallow sequencing, you're still using paired end mode, and it's usually shallow sequence on a MiSeq or a mini-seq platform. So you have that option as well as a QC checkpoint before deep sequencing. Could you show more results? What would you get in each method? Could you get 3D genome image and some sequences data? Yep, that is a great question. I'm thinking here we do have some, our technical notes that they would highlight for each method, kind of what is the information that you're getting. I try to distill down in this presentation, but we can definitely share with you these tech notes that give you for each product a little bit more of a visualization of what kind of data you're going to get and how you're going to visualize that. So we'll follow up with you on this. We do have the email address captured, and we'll follow up with you on more of that documentation. Can we use TFS promoter interactions for plants at all? So the question here is a little bit, yes, you can, but which is the right assay for you in the sense it's going to just be taking also in account, the sample type that you have, which in the case is plant. So I've seen, both sides of how that implemented. One way for people that are usually interested in really specific, in a specific transcription factor, you can still run Hi-C. It's not validated for plants, but you're looking here at understanding how the micro-C prep works for the plant, and you're looking at, using your antibody for that selection. So in terms of the first part, you are going to get a micro-C guidance in terms of how you prepare the library prep, because this is something that we have worked on and customers have worked on for plants. So the Hi-C proximity ligation of this equation is something that would be easily implemented because we do have the experience of running the assay on plants. The antibody, then it's going to be a little bit more of, how are you using the antibody that's ShipSeq validated? Do you have that antibody available? You're going to do a little bit more of that, titration to get to the right amount of antibody and lysate input that you're going to get more of that high quality library, so high complexity library. So to address your question, it's doable. We're happy to chat with you about which route you'd like to take when it comes to that. If it's a service or a kit, it's definitely doable, but you will need, a little bit more of that, trial and error when you're running this prep, just because it's not something that we have validated. What are some considerations we should take into account for primary patient samples that might be more fragile and difficult to work with than cell lines? Are there certain kits that might work better, or any alterations to the protocol that you would recommend? This is, again, a great question. So with this, if you're interested in, for example, micro-C for, these patient samples, or even TOPOLINK, I think we do want to facilitate you still getting the right assay for your research. So, just because you're working on samples that are more fragile, you wouldn't want to compromise on the assay that is right for you. What I've seen with some of those fragile samples or cells is more that there's a tendency to cross-link them, before you freeze them. So our current workflow, you harvest your cells, you wash them, you freeze around 1 million cells that you would like to start with. Then you take that sample out, you cross-link with DSG, you cross-link with formaldehyde, and then you continue with the digestion. For samples that are more fragile than others, or samples that would undergo, not even only patient samples, but samples that would undergo some pressure, a FACS sorting, what I've seen researchers do is just cross-link these samples with formaldehyde, before they freeze them. And then that way, when they're getting them out of the freezer, they're less worried about them, lysing while freezing or the quality dropping. And then you can take those cells. You would still need to do the DSG cross-linking, because this actually using DSG in combination with formaldehyde will help capture more longer range information in your library. And this is something that we use for most of our workflows. We actually use this DSG and formaldehyde cross-linking. So you would do the DSG cross-linking, but you will just skip the formaldehyde cross-linking, and you can proceed on with the workflow. So I think another question was about the antibodies. Do we have antibodies that we can work with or need to get your own? I think we addressed this question with, it's user supplied, but we have a list of things that we validated to help the work on, you having to validate those antibodies. So you can just plug them in the workflow and use them. And if we were working with us through services, not kits, that would be something that we will be using, unless you have an antibody that's specific to you, something that we've not validated, then we'll work with you to define and validate that antibody. We still, I think, have time for one more question, and then we'll address any question that was not addressed, today in real time. We will also address them offline. So I have the email addresses for everybody who has asked a question, and I'll be able to reply to you offline. To follow up on your ATX coverage for 5 kb resolution for a human with your pipeline, how many loops or tabs you expect to call to judge you have a successful library? So in terms of judging that is, a successful library, we don't really base that on the number of loops or tabs that you can call, because these numbers, depend also on your sample biology. Are you expecting to have, for example, for high chip, are you expecting to have more acetylated profile? If you're using the HGK27 acetylation antibody, are you expecting to have, lower acetylation profile? So we don't base it as much on the number of loops or tabs. That said, we do have in our tech notes that I can share, some indication of the ranges instead of like specific values. We have ranges that we usually see that we can get to with that resolution. So we can share that with you. But in terms of, understanding that if you have a successful library, if you have, a library that you're able to QC, whether you've done that QC, before as a shallow sequence library, or if you have already deep sequenced your library, you can always check the quality based on the long range information that you have and the duplication rate that you have. So essentially, our expectation is if you have a library that has good long range information and low duplication rate, or a duplication rate within what is expected from a Hi-C method, you should be good to go with your analysis. And then the question becomes more about, your sample biology, the resolution that you're using. So do you need more data for that resolution? One thing we also do that has worked well is, for example, if you're trying to call TADs, what we do is you would call TADs at different resolutions, and then you merge these TADs together. It increases the number that you're calling, because what happens then is there are, some things that you would see at a higher resolution, but there are things that you actually need a lower resolution to see. So for example, for TADs, what we do is we start with a 25 or even a 50 kb resolution, then we call TAD as a 10 kb resolution, and you can merge the TADs that are called at each of those resolutions, and you can remove anything that is, duplicated, but then you get a higher number of TADs, because also TAD has a hierarchy structure. So you have your TADs and sub-TADs, and you're able to call each of those at a different feature, at a different resolution. And in the tech notes, we actually have an indication about, what to expect from a certain resolution. So we can share that with you. I think also what would be helpful, maybe Jesse, is as we're sharing this recording with our attendees, we can add links to, our website or the product pages on our website that have these tech notes with the information that were requested. So that's something we can think about.
Yeah, that's a great idea, Miriam. Thanks so much for the presentation. Thank you so much to everyone who joined us today. We really appreciate it, and we love connecting with you, our community, and I look forward to doing it again. Miriam will be back again very soon, no doubt, with addressing other technical issues or technical questions you may have. You can always reach out to us and let us know proactively what you're interested in hearing about, and we will bring those webinars to you throughout the year. And as Miriam said, I'll be sending the recording out, and we'll also link to some of those documents that she mentioned during her talk. And we've also got your email addresses. And if you have any questions, we will follow up with those that weren't answered today. Thanks so much, everyone. Take care now.